It replicates, takes over the DNA, as we move on, because a lot of things that are held constant in biology do change. This doesn't change. Oh, sorry. General biology does not change. That is very recommended. You guys are on more of an inductive path. Dehydration synthesis. You may not need to copy that. Condensation reactions. Dehydration synthesis is, shout it out, the addition or subtraction of water. Subtraction. Dehydrate, to take away water, to put molecules together. Okay? Let's see who knows their stuff. Okay? If this was honors biology, this would all be plus points. But I can't give you plus points because college doesn't work that way. I know. You cannot get extra credit points in college. At least I never did. The question I have for you is a dehydration synthesis reaction that connects glucose to glucose to glucose to glucose, making a polysaccharide, has bonds that are called what? Yeah, Shell. Glycosidic bonds or glycosidic linkages. Very good. And today it's going to get very, very intricate at the structural function of these complex carbohydrates, as well as there's going to be one thing you're going to have to memorize. You must memorize the shape of glucose. Is glucose in a ring or is it linear?
Ring.
It is in a ring. Needs enzymes. How many enzymes need to break down various, or to build it up and to break down? How many enzymes? A ton. Hydrolysis is the addition of water to then break down. So this is a little bit of a review. Where does digestion start? And this goes to the anatomy kids. You may know this. It starts digestion in the mouth. In the mouth. Polysaccharides can be broken down into smaller, very large polysaccharides, and monosaccharides and disaccharides can be immediately absorbed into the bloodstream. Not all disaccharides, some. Simple glucose, and yes, Rochelle, galactose, and fructose can easily be just put right into the bloodstream. Okay? Wonderful. They're mowing the lawn during the school day. I'm going to get in trouble today. Yep. I've already got in trouble for the door. The door still opens up for the third year in a row. And I said something sarcastic that. All right. Digestion. Starch is hydrolyzed in the duodenum. What is the duodenum? It is the. Okay.
First 25 centimeters.
First 25 centimeters of the small intestines. Okay. Most of your food, most, not all, most, is absorbed and, not absorbed, is broken down, hydrolyzed in the duodenum. It is then absorbed by these little, very, they have a very, very small structures that look like cilia. They're called villi or microvilli. And their job makes the surface area very large, increasing that surface area allows for more absorption. If we could take our small intestines, all 25 feet of it, and spread it out, okay, it would go from here to the back of the room. One big long chain. But if then we started to stretch it out, and then we took the villi and stretched it out, it would cover this whole room, plus. The lymphatic system is your other blood circulatory system. It cleans your blood. Glucose. We extract energy out of this molecule during cellular respiration. There's your first term of cellular respiration. The carbon skeleton, and this comes out of Campbell's, serves as actually, they call the raw material, which basically means the CH-CH-CH bonds of a carbohydrate can be used for lipid and protein buildup during cellular respiration. Okay. The rest is used to create dipolysaccharides. You must be able to do this. That's incorrect. Hard drawing a hexagon. And that's incorrect. You keep writing, I'll get it. Don't mind me. Yeah! Woo! And the reason why I'm having you do this is very important, and I'll explain why. That should be the O. Someone check me out and make sure I'm right.
I thought the OH went on the third.
Yeah, I thought that was on the third.
Yeah, that was the
No.
No, I thought it was on.
Did I have it right the first time?
I thought you did.
That's what I found right.
That's what I had.
You got it.
Someone verify that for me.
I put a squeeze out
We have it right.
Yeah.
Yeah.
It is
OH.
on the third one. So you're right, that's right. That's
And what I had first was right. Good. Okay. Okay. This is glucose, and you need to memorize that. The reason why comes later. So we do need to memorize glucose. You do not have to memorize fructose. You do not have to memorize galactose. In fact, around the third one, 1, 2, 3, if I switch these two, that would be galactose.
Dun dun.
Well, I don't understand what these notes mean.
What notes?
That the carbon skeleton is the raw material.
This can be used. These can get broken down through hydrolysis and can be used for the development of making lipids and proteins and nucleic acids in your body. They help form other organic
So by
macromolecules.
breaking down that ring, we.
Oh yeah.
Okay, and then that oxygen is in the ring, so it's not just all
It's
carbon
not so much breaking down glucose,
as it is any
it's
carbon.
the carbon skeleton of a carbohydrate. So it's taking large chunks of the CCC, the hydrocarbon part of it, can be used for the making of lipids and for fuel resource. That's why it's called long-term energy. There's your disaccharides as a review. Glucose equals maltose. Glucose galactose makes lactose, and glucose fructose makes sucrose. Okay? All formed with glycosidic linkages. What is maltose? It's good for fermentation. When you get to be 21, you can enjoy beer.
You can't enjoy it before then. You just hate it.
You can enjoy it with your parents' permission in their presence. Or you can bring it to class. I don't really care. Next, sucrose can be used for, once again, simple table sugar. It can be found in fruit. It can be found in candies. But it also can be found as being transported through xylem and phloem up through adhesion and cohesion to the tops of leaves, or the pinnacle of leaves, up through the roots to leaves. Can also be transported down. lactose milk, hence lactose intolerance, which are various levels. Anyone lactose tolerant? Excuse me, lactose intolerant?
So like a
Okay.
whole
It doesn't necessarily. Can you drink any diet? Can you have any dairy products, Ebo?
Like, I can, but
Okay.
I'll have tummy problems.
Oh, you do have problems. You have a pretty extreme case then. You know, there is a way for, I believe, there is stuff out there that can make you lactose tolerant. Your body can change. I don't know. I think it can be, I don't know. There can be pills taken to help that out. Okay, here we go. Polysaccharide. Well, first of all, an oligosaccharide, there's a term for you. O-L-I-G-O, then saccharide. I don't know if that's in your notes or in your book.
It was last year.
Yeah.
Oligo-6 is between 3 to 7, 3 to 8. It's no definite number, 3 to 7, 3 to 8, 3 to 9. Is an oligosaccharide. It's a tweener. Yes, you need to know the term oligosaccharide. They don't really have a great amount of functions. They're still easily broken down. It's still very short-term energy. But as you can see here, amylose and amylopectin. Starch is made of those two. Maybe you can see that better. No, probably not. The Campbell's book is magnificent about showing you these differences. Now, what on God's green earth do I mean by a 1 to 4 glycosidic link? Not only do you have to know glucose, check her out, and that is glucose number 1. This is carbon number 1, carbon number 2, carbon number 3, 4, 5, 6er. When I talk about a 1 to 4 linkage, this is going to hook up with another carbohydrate. Well, actually, it technically would look like that.
So it was like the 5 to
There we go. And this would be the 4th carbon, 4 to 1, 1 to 4. Okay. So when we talk about the glycosidic linkage, it's the standard 1 to 4, carbon 1 to carbon 4. 1 to 4. This is another glucose. Yes.
What is the difference between an oligosaccharide and a polysaccharide?
Oligo is just, it's a branch of glucose molecules together from 3 to 7, 3 to 8. Think of it as a immature polysaccharide. All right. What is starch? Starch is amylose, and it's amylopectin. A starch is about 20% amylose, 80% amylopectin. Some foods have more amylose than they do amylopectin. An example of that would be corn. Corn has way more amylose than the amylopectin. But if you look at a potato, a tuber, that has way more amylopectin than to amylose. Okay? So, the relationship is 20 to 80. What is the difference between amylose and amylopectin? If you look at amylose, it is unbranched. It is simply 1 to 4 linkages, without being branched. Hence linear.
What does branched mean?
We'll get to it. Amylopectin is branched. Branched means no more 1 to 4. Branched means carbon 1 attaches to carbon 6. So that means if I were to draw this ruler here, this would be a shape of amylose. It is linear. If I wanted to have it branch, I would branch it off now, and now I've created a branch. It is now not linear. This branch right here, all of these connections between the centimeters would be 1, 4, but when we started to bring this ruler in, the branch would be 1, 6. Yes.
So amylopectin has both 1, 4 and 1 to 6.
Yes, when it branches.
Okay.
only at the branches at 1/6.